Hi, good afternoon, everyone. I'm Rafael Eugenio from the University of the Philippines, Diliman. So, so I'll be discussing this afternoon. I hope you're still with me. The presentation, Enhancing the UPCC Yeast Identification, Insights from the two markers, ITS14 and D1D2 gene markers. Here, some of my co-authors, Paul Christian Gloria, Joshua Veluz, and Dr. Siringan or Dr. Oxy. So unlike the other culture collections that we've heard earlier, so we're considered as a small culture collection preserving the three like bacteria, yeast, and filamentous fungi from various sources. Right now, the UPCC has at least 108 accession bacteria, 88 filamentous fungi, and 88 yeast as well. So these are some of the current isolates that we're still authenticating and checking for activities. There are 37 bacteria, 22 yeast, and 10 filamentous fungi from local sources. However, even with the effect of the pandemic on us and the retirement of some of our staff, we have a similar situation with Dr. Zunita that the skilled personnel leaving the culture collection has a big impact in our culture collection. So some of our cultures were um, actually gone from I don't know what happened because of the pandemic and because of the lockdowns in the Philippines. So we're down to those numbers from the previous slides, but we also have some efforts to increase the cultural holdings through research projects. So we have here the isolation of bacteria with plant growth promoting potential. These are isolates from the province of Isabela and Cagayan, which were isolated from the rhizosphere of bamboo and also the local corn in the Philippines. Some were also isolated for fungi. We had some isolates that were isolated from Senna elata. This is a common plant in the Philippines. And then we just wondered what if there are other fungal isolates that we can use for their biocolorant potential. And lastly, this is where I come in. Since we're working on bacteria and actually yeast from local fermented products for probiotic and psychobiotic properties. So for those of you who are not familiar with psychobiotic properties, so it has been, I think there are still debates with the gut-brain axis in using those probiotics to have some effects with some mental health illnesses like anxiety and depression. So that's what we're trying to look into for the bacteria and yeast isolates that we have from local sources of fermented food products and beverages. Here are some of the commonly identified bacterial genera, and as we can see, it's usually the bacteria for probiotic candidates, but searching the literature, we only have Saccharomyces, and that Saccharomyces is just Saccharomyces boulardii, which is a commercially available yeast and has been discovered for quite a long time now. But it can also, it is beneficial in terms of the following. It can detoxify your mycotoxins by binding to those identified mycotoxins, and then the bioavailability by degrading the phytic acid, and of course prevention of intestinal diseases by the actions of their co-aggregation. And some research groups in the Philippines were also trying to check the viability of yeast such as Saccharomyces in being a vehicle for some RNA vaccines. Okay, so we've checked the efforts on screening probiotic yeasts, and these are the international journals. They've checked the kombucha, also some Brazilian fermented olives. In the Philippines, basically, it's still focused on the lactic acid bacteria, very, very minimal on the yeast part. Also this one with Malilay et al. Okay, so this is just to show you the number of publications that are related to either probiotics and probiotic yeast. So as you can see, when they had this review and they've checked the papers from 2015 to 2020, Probiotic yeast is very, very far. So there's like thousands for bacteria. But when they filter this out, it's just less than 500 for those papers, even until 2020. So we're trying to contribute and identify um, those bile salt tolerant and acid tolerant yeasts from within the UP culture collection. So some of those were sourced from these decaying fruit, some were sourced from local rice wine, and some were obtained from fermented fish, fermented rice. Okay, so um, once we've screened those for the tolerance assays, we also wanted to identify them through molecular means with the two markers that were given earlier. That's ITS14 and the D1D2 markers. And here is just a brief overview of the methodology for this project. We've just separated here the types of medium that were used in the maintenance of the yeasts. The picture here is a 96 well plate since we did the tolerance assays in the 96 well plate since we don't really want to have a lot of plates for your samples and also to minimize the funding for the tolerance assay. And then we had all of the yeasts with and without the tolerance showing in the results. We had them, all of the isolates had the DNA extraction and then a PCR for the amplicons of ITS1, 4, and D1, D2. So those were sent to, I think it's in Macrogen here in Korea for sequencing, and then we did the phylogenetic analysis afterwards. Here are just some of the software and the two of them are software, and this one is a command line to have your phylogenetic tree and model testing. So for the results, we just identified here, I'm just focusing, we had a lot of samples, so I'm just focusing here on those with the activity. So our target is actually between pH 1 to 3, since that was the ideal pH for the gut. But we just had some activity on pH 3 usually here in 2145, 2160, and 2172. So this is for the first type of medium. The second type of medium had only one activity for pH 2. That's relative to the unadjusted medium here. Okay, this one's on the bile salt tolerance. I think the ideal tolerance test would just be around 0.3% to 0.5%. But as you can see, even with the heightened levels of bile salt, the yeast isolates were able to surpass those numbers in the unadjusted medium. And I don't know, Dr. Oxy was telling me that maybe I should test them. Maybe they want to have the environment with a higher bile salt. So I don't know, that might be the next phase for the project if we have the funding. So these are just the isolates here that we were identified to have the bile salt tolerance after 48 hours. Okay, for the YMB isolates, we only have around 3 isolates showing bile salt tolerance. So it's 2032, 2065, and 2067. Okay, proceeding now with the ITS14 sequencing and analysis. I can't show you all of the phylogenetic trees, so we just had a selected number of UPCC isolates, and we were able to have a putative identification using just one of the markers. This was previously identified Candida cruzae in Isachenka orientalis, and we were able to have a convincing number here for its identification having Pichia kudryavtsevi, ATCC 6258. So this was inferred using 465 nucleotide positions. And this is the best fit model for this particular analysis. The next isolate would be 2145. This is also the acid and bile salt tolerant isolate that we had. But as we can see here, the Hansenula anomala and weaker Hamomyces anomalous based on literature were just synonyms. But here in this circular tree, it's not really that convincing even for us. So we wanted to do some further tests on this, but that would depend if we can move further with the probably like whole genome sequencing of the isolates. It, just setting here an example as well for UPCC 2145, we've also tried to do a haplotype network analysis. And with the haplotype network analysis, we just considered all of the isolates reported from the same source. So most of the reported isolates here are just from rice wine from different countries, and we just group them on East Asia, Southern Europe, South Asia and Southeast Asia, and Eastern Europe and Oceania. And here we can find that the isolate that we have in the lab is just related to one isolate in East Asia. We're still not sure why this has happened because we're expecting that the isolate would be related to most of the Asian isolates, but somehow it's not showing that here in haplotype network analysis. This one, UPCC 2072, did not have any good activity on the acid and bile salt tolerance tests earlier, but we were able to get kind of high bootstrap value and model here for the UPCC 2072, identifying it as Yaruia lipolytica using the ITS14 sequence and also the phylogenetic analysis. Here we also use the other marker, D1, D2, to identify UPCC 2072, and we were able to get Yaruia lipolytica as well. I'm sorry for the figures. This was better on the once we generated this, but I don't know why. What happened here? Okay, so um, we compared the identification that we had based on the NCBI blast and also the Unite database. If you're not familiar with the Unite database, it's just a curated database of the ITS sequences based also on blast, but these ones are validated by scientists both in Europe and the US, and we were able to check that some of our isolates did not have consistent identification, like our Candida tropicalis, the three isolates that we had were identified under a different identification, Oligophagozyma castelli. I think the synonym of this identification is Candida castelli, so we're not sure if we're going to retain this one or the other identification. We need to do further tests or further sequencing for these types of isolates. However, for the others, we had consistent identification, so we have no problem with that. And then comparing the two types of markers that we used, basically, we still had consistent naming except for the 2062. So we'll still have to check this isolate now after probably if we get some more funding. Okay, we also tried to check if we can do whole genome sequencing in yeast. So here, we were quite successful in getting enough DNA and sending them for whole genome sequencing using the Illumina Nova 6000. And these are our values. We use at least four different types of assemblers, and these were our results. These are the best numbers that we got with the largest contig of 992 and 773 base pairs, and then 50 of 233 and 220 base pairs. Kind of low GC content, but we're thinking probably this is just enough. And then the Busco completeness is just 96.30. So we're thinking if we can do hybrid sequencing, long reads plus the short reads in the future. Yeah. Okay, and then after doing the genome assembly of that isolate 2072, we tried to identify if they have some of the biosynthetic gene clusters using the online platform. It's Fungi Smash. And using this platform, we were able to identify some BGCs that had that were related to terpene production. However, we must take note that the similarity score is very low. That's just 0.38. And this is similar to what they got for Aspergillus. We were not surprised then because according to literature, some Yaruia lipolytica yeast were also being used for terpene production through genetic engineering. But probably in the future, we can get more of a similarity or might be a different result. Another BGC that was um, gathered from the fungi smash is on the NRPS, and it's also truncated here with a similarity score of just 0.20. This one now is related to its symbiont role in the burying beetle. So I guess that's just the bulk of the results, and this is just the summary and the future directions for these potentially probiotic yeast that we had at UPCC. So it just highlights the utility and the contribution of the microbial culture collection in the discovery of potentially probiotic yeast isolates. At least 5 of the isolates exhibited or more than 5 of the isolates had good acid and bile salt tolerance against the unadjusted medium. And we also wanted to highlight the significance of those molecular methods, the D1, D2, and ITS14 gene markers for putative identification. But we are still, I'm highly inclined to do the genomics, probably for mining the biosynthetic gene clusters. We might find something else there in their genome after we do the assembly. These are just some of the future directions of the research. We have to do the safety assessment, of course, more genome sequencing, and then bioinformatics analysis. Would just like to thank my laboratory, the Microbiological Research and Services Laboratory. The MRSL, or the Microbiological Research and Services Laboratory, actually hosts the UPNSRI Culture Collection. And this study is made possible by the Office of the Vice Chancellor for Research and Development of UP Diliman. And this is just the title of the study. So thank you very much for listening.
Thank you.